I'm very proud to be up here today. I'm David Fontana, the Chief Business Officer and Chief Operating Officer. I'm really proud to be up here on behalf of the 150 Umojians back at work, either in Boulder, Colorado, at our manufacturing facility, or in Seattle, where our R&D headquarters are. Umoja is a leader in vivo cell delivery, cell CAR T delivery. And today I hope to share a little bit with you who we are, what we're about, and then where we're headed. Our mission is pretty straightforward, but it's an ambitious one. Really, based on the decades of success of CAR T therapy, we really want to improve patient access and impact of the CAR T therapies and those indications that are out there and approved, but also new indications like solid tumors. I want to highlight the word access, and I'll show you why in the next couple slides. Today we have beautiful CAR Ts out there. There's 6 CAR Ts on the market for BCMA multiple myeloma and CD19 for lymphomas and leukemias. But the limitations of these CAR Ts, which I'll show you, really are only meaning half of the, or a fraction of the patients are getting these CAR T therapies. I had the honor to be the global project lead for a product called Breyanzi through the Juno, Celgene, Bristol Myers Squibb, from the first patient all the way to the end. And this is some of the data I'm proud of at the bottom here. But if you think back in 2017, a patient who had large B-cell lymphoma, a very aggressive disease, really had little possibilities. And you see on the top bullet here, there was this overall 2-year survival rate of 20%. They were getting just garbage chemo, salvage chemo, and mostly hospice-type drugs there. Play forward in 2017, Kite and Kariah come out. All of a sudden, patients are getting cures. There's patients that are cures. We have patients beyond 10 years now that receive those drugs that are still disease-free. I don't recall anywhere in this field there's been such transformational data. I'll brag a little bit on our data on the bottom here, the Breyanzi data. I have nothing to do with Bristol Myers Squibb anymore, by the way. I'm proud of this data. This is looking at patients who failed R-CHOP, and then the standard of care for over 30 years was a big dose of salvage chemos, and then stem cell transplant. Standard transplant, everyone who went through this, if you're eligible for that. This graph shows a CAR T study called Transform, where if you just look at the PFS data, 21% of those patients had, only 20% of those patients were progression-free at 18 months versus 50% of one dose of the single CAR. Wonderful data. There's even better data with BCMA these days. You can see out there with Carvykti and Abecma, and great data. The problem really comes is that patients will relapse, and even in the top nations, US, Europe, and Japan, less than 30% of the patients that could be getting these therapies are getting them. And that's what we're about. That's why when I said access, that's what we want to solve. And why is that? So first of all, I think we've heard maybe 12.5 presentations in the last 2 days about the limitations and complexities of manufacturing. And I do give applause to all the CDMOs and consultants that helped and stepped in. Small companies and even big companies can't struggle with this. Bristol and J&J still struggle with the supply chains for these things. The difficulty in the administration process, all of these require lymphodepletion. If you don't get rid of those B cells and provide that cytokine stimulatory agents, they're short-lived. So patients who get CAR Ts for oncology have to get lymphodepletion for that exact reason. The footprint's limited. The FDA has given permission in the US for 125 treatment centers. So if you're sitting in beautiful Walla Walla, Washington, in the wine country, you've got to pick your family up, yourself up, leave your church, your support network, and move to Seattle for 2 months to get your CAR T therapy. Not a real great experience for patients. Finally, the complexity of taking your cells out in Walla Walla, processing them, shipping them to Bothell, Seattle, getting them manufactured over a month, and hopefully getting them back in a timely manner, and then go through all the process, as I mentioned, is really difficult. And that's really what we're about. We're excited and extremely proud of all the CAR T data out there, but think there's some room for improvement. What does that mean then? So, all these great data, these limitations, it means that the slot times for patient waits in some of the key hospitals are months and months. You probably read some of the pretty heartfelt articles by physicians saying they have to play God, where there's 20 patients waiting for a Abecma or for Carvykti, and they get 2 slots a month. What a horrible situation, right? And that's what I mean by access. months and months wait, and then you get failures. If you look at the labels of these drugs, it's just to be transparent. You wait those 3 months. You're one of the lucky ones that get picked, and at the end of the day, you don't get your car because of the manufacturing. Some of these products have 30% manufacturing failure rates. So all that process, all that wait, you're selected and you're still failing. And then a lot of times you go through all that, the patient conditions are just too acute. Aggressive large B cells are very acute disease. ALL in the children is a very acute, fast-acting disease. And so that's what we're about. We're trying to solve for these problems. So great that I say all that. How are we going to do that? I'm not going to go too deep into our science, but our platform we're going to talk about today mainly is our in vivo gene delivery platform. It's called VivoVec. It's a third-generation linear vector trafficking product. Basically, we make these VivoVec particles. Our secret sauce is really how we surface engineer for tropism and for activation, and really trying to replicate the much we can is the antigen-presenting cell T-cell interaction to get activation and robust transduction of these cells. So these particles are manufactured, and they include all of the contents needed to transduce effectively, permanently too, because it's lenti, permanently the target T cells in the patients. So the CARs we can put in there, as I mentioned, CD19 is an obvious one. We could put a CD19 CAR in there and go and then provide benefit for the patients with lymphoma. So I mentioned too lymphodepletion. So we wanted to get rid of a lymphodepletion. So what's the deal? How are you going to do that? You can't give in vivo CARs with lymphodepletion, right? So our solution to that is something we call RACER, rapamycin activated cytokine receptor. Really nifty technology where we basically took the heads of IL-2 and IL-15, knocked them off, and replaced them with the heads from a drug called rapamycin. So when these cells, or when these receptors see rapamycin, they basically produce the exact signal that an IL-2 and IL-15 would be. What's nice about this is they do it only in the engineered cells. So I'll go through this one more time. So we give these VivoVec particles. The particles are the drug product. Put those in the patients. They contain at least 2 payloads. One's the CAR payload, CD19 we'll use for an example, and the RACER payload. The drug is in the patient. We get an activation when the CD19 sees the tumor. We get an activation, expansion of the T cells, and then we can stimulate more growth of those cells by giving rapamycin systemically. Quite an elegant system, we believe. Well, the question is, does this stuff work? Well, here's our data that we're going to show. It's our non-human primate data. It's data on our product called VV300, which is a CD20 CAR. This is done in a very particular non-human primate model. There's great benchmarks with the CD20 autologous CAR with lymphodepletion by one of our co-founders, Dr. Mike Jensen. So in this study, we're basically looking at the first 4 monkeys we dosed on the bottom there. I'll go through some of the highlights. The first monkey was the lowest dose we tested. Just to give you orientation, the first column is the pre-injection baseline levels, and then this graphic only goes out to day 14. I didn't want everyone to have to look at the eye charts out to day 70, but then you see the bottom 3 monkeys are at our clinically projected dose. The little red dots in the there represent the CD20 in the animals, and then the blue flow diagrams represent the CD20 CAR. And what you can see nicely, even in the top monkey, we didn't generate a ton of CARs, but there's a little bit of CARs at the low dose and some mild degree of B-cell depletion. What's amazing to us, and amazing to me in particular, is if you look out at day 10, you can see full B-cell depletion in all the monkeys, just no cells to be found, and a generation of CARs from 27% up to 65%. So 65% of that animal's T cells are CD20 positive CARs. So I would have never imagined in my life that you could give a systemic little particle and generate not only CARs, but then generate this kind of activity. So really, really excited. What I don't show on the slides here is even some more interesting data. Once the B cells go away, the CARs are quiescent. So then eventually the B cells come back. What we've been seeing now is that up to day 70, day 73, when the B cells are starting to come back, we're seeing a response again. So there's some residual cells, and they look like to be memory cells now, that then regenerate and kill the CAR Ts. And this is done without rapamycin. So what we're excited about our animal data, and we do believe it's the best in class, is that this is very comparable to lymphodepletion, CD20 ex vivo autologous CAR data shown in the exact same model. We're really excited about that. We haven't even got around to testing our rapamycin system yet, so quite excited about our data in this cell, which drives our pipeline. I talked about VV300, that's the CD20 CAR, but we also have a VV111, CD19 obviously, car, and that'll be in the clinic next year. And the technology I'm not going to talk about. It's just too complex, but we're really excited about it. The VV200. This is basically, we use VivoVec in the racer system again, but the car for the VV200 is a universal car. It's an anti-FTSE car, and these tumor tags are small molecule binders that you can have a bunch of them. And we have one called 170 that's in the clinic already, which is for folate. So you can give a cocktail of these tumor tags along with VivoVec and basically produce responses in a lot of different tumors. So we're quite excited about our pipeline and the growth of our pipeline. So what does this mean? I like this slide here because it really shows how simplistic this is going to get for us. We have now an engine to be able to put anyone's car, whether it's our cars, the CD20 and 19 as I mentioned, or on the bottom there it's our tag car, or anybody else's car, BCMA or WER1, solid tumor cars, or 33, into our VivoVec system plus racer and generate in vivo cars with the ability without lymphodepletion to stimulate those cells and grow them in the patient. I really like that example because you can imagine the treatment paradigm. If a patient doesn't need rapamycin, if the tumor is killed and there's plenty of CARs, you wouldn't give rapamycin to expand them. If a patient has a PR or a waning CR, you give a couple doses of rapamycin, can stimulate those CAR Ts again, kill the tumor. So it's a really predictable and controllable way to do it versus lymphodepletion, which is a hammer at day one. I won't talk about much at the bottom, but it's the same thing. If somebody can build a small molecule or a small protein tag, you can use a host of those tags with one single CAR. And that's what we're really kind of our next technology we're going to tackle here. I forgot to mention in the beginning, and it's an important thing to say, Umoja was founded by a bunch of great scientists who've been in the industry and have a lot of success. The name was coined Umoja, which means unity, like bringing things together. The idea was bringing all these technologies together, but also bringing people together in the company and bringing partnerships together. We formed a lot of early partnerships that have just been fantastic. We learned from our partners everything from Seattle Children's helping us with clinical trials through an IIT. We've got agreements with other companies as well to do different things with our other platform that I'm not talking about, the iPSC platform we have that uses the racer system as well. And we're hoping in 2024, based on our success, that we'll have some big pharma deals to announce. So hopefully you're rooting for us. One of the investments we made early on, and this is a big controversial thing at the time when money was free and we could all write checks, was we decided to build our own manufacturing plant in Boulder, Colorado. For all those reasons, we thought as a little company and one who was really interested in partnering, if we control the supply chain from day one till the end, we would have an advantage. So after a grueling 18 months of criticism for spending that money, we finally opened up the manufacturing plant 2 weeks ago and are starting our first engineering run actually this Sunday. So we're quite proud of that. Beautiful facility, but it enables us to make all the antiviral vector particles on a clinical scale. One batch, 200 liter, can make a couple hundred doses for patients. We've got a whole football field yet to build out for commercial scale when we go do so, but we're quite excited about that. What we like about our facility too is right now we can use the process development people who then can move across into the same building and help with the manufacturing. They can move on to the quality and release testing, and we package it manufacturing warehouse all in nicely done in Boulder, Colorado. Great place to visit. And we're fully loaded and ready to start support our clinical trials and partner trials in 2024. The team. Very talented team. We have a lot of folks that came from cell therapy backgrounds and actually true manufacturing backgrounds. A lot of us are from the Juno, Celgene, BLMS lineage, others from Lyell, even back to Dendrian days, part of our crew is. I want to highlight two characters on this screen though. Dr. Byung Yao, he's our head of vector biology. He's the one that's doing all the great surface engineering and just killing it, making there. He's going to be presenting. We got accepted for an oral presentation on our next set of non-human primate data at ASH. So he's going to be killing it there at ASH. And then I want to highlight my co-founder and best friend, Dr. Ryan Chrisman, because he's not only our CTO, he was the one that stuck his neck out, and he lives in Denver, so he was the one who stuck his neck out, built the climb. It was also his birthday this week, so he had to leave early here. And he was also the 2023 Mesa golf champion. He had the longest drive as well, so congratulations, Ryan. I think I've never seen him as proud. I think he's more proud about that than the climb, but leave it for another day. Yeah, great stuff. Highlights for this year, I showed you the NHP data. Again, it's the best in class. No one else has generated CARs in the plasma like we've generated. Simple as that. We have catalyst events where we're taking our VV200 to the FDA December 1st. We've got a meeting there, and we're locked up and loaded to file our IND in the first half of 20 next year. I better hurry up. I want to close on one last thing. Our theme is patients are waiting, and that's really the theme. And you saw what I said. Patients are waiting for the therapies. When I signed up for Umoja, one of the first things that was said to me is patients in Mexico get cancer too. None of these CAR Ts are approved there, and that's what we're really hoping to democratize cell therapy and give those great transformational efficacy to a lot more patients. Thanks for your time.